I am CH Vidya, Assistant Professor of Biochemistry from Government City College, Hyderabad, and today I am going to deal about the topic behavior of amphipathic lipids in water, formation of micelles, bilayers, vesicles, and liposomes, and another topic fluid mosaic model. So let's look into the topic behavior of amphipathic lipids. Molecules which contain both hydrophobic and hydrophilic groups are known as amphipathic. So we already learned in our previous videos that lipids are amphiphilic in nature, that is they have hydrophobic character as well as hydrophilic character, weakly amphiphilic, right? So molecules which contain hydrophobic and hydrophilic groups are called as amphipathic, right? Examples are among the lipids, fatty acids, phospholipids, sphingolipids, bile salts, and cholesterol, and that to some extent are amphipathic in nature. So among lipids, these are the ones which have this amphipathic nature, that is they are hydrophilic as well as hydrophobic groups are present in them. Phospholipids have a hydrophilic head. Hydrophilic head. What do you mean by hydrophilic? Hydrophilic is water showing affinity towards water, water attracting. So they are called hydrophilic. Hydrophobic is water repelling. They do not show attraction towards water. They repel water. So the groups which repel water are hydrophobic. Groups which attract water are hydrophilic, or which get attracted to water are called as hydrophilic. Right? So in phospholipids, the hydrophilic head is phosphate group attached to nitrogenous base. So the nitrogenous base could be choline, ethanol, amine, inositol, anything. So the phosphate group attached to nitrogenous base is called your hydrophilic head. And phospholipids have a long hydrophobic tail. The hydrophobic tail is nothing but the glycerol and the fatty acids attached, right? Next, the general structure of an amphipathic lipid may be represented as a polar or hydrophilic head with a non-polar or hydrophobic tail. So how do you show the structure? Now if you see in this slide, you have the structure of fatty acid. See, you have CH3, CH2 whole N, COO minus, right? The hydrophobic nature is due to that tail region, that is CH3, CH2 moiety, whatever is present, that is hydrophobic in nature, whereas the carboxyl group is hydrophilic in nature. So fatty acid has two groups, hydrophobic as well as hydrophilic. Now you have to say which is hydrophobic, which is hydrophilic. The hydrocarbon chain is hydrophobic and the carboxyl group is hydrophilic, right? Similarly, in the next picture you see, that is the picture of phospholipid. Okay, phospholipid, and that is phosphatidylcholine, right? So you have the glycerol, to the glycerol two fatty acid chains attached. So that glycerol with the fatty acid chain attached, two fatty acids attached, that is hydrophobic in nature, whereas phosphate and choline, you can see it is differentiated with pink color, right? So phosphate and choline are hydrophilic head groups. In this way, fatty acids, phospholipids, if you take cholesterol, cholesterol has a hydroxyl group, right, at the third position. So that hydroxyl group is hydrophilic, remaining ring structure is hydrophobic. So in that way, these lipids have hydrophilic component as well as hydrophobic component. That is the reason the lipids are amphipathic molecules. Next, fatty acids contain a hydrocarbon chain with a carboxyl group at physiological pH. The carboxyl group is polar in nature with affinity to water, that is hydrophilic, while hydrocarbon chain of fatty acid is hydrophobic, right? Next, orientation of amphipathic lipids. When amphipathic lipids are mixed in water, so when such lipids which are having both hydrophilic and hydrophobic components are mixed in water, that is aqueous phase, what happens? The polar groups or the hydrophilic groups orient themselves towards aqueous phase, while the non-polar tails orient them towards the opposite direction, that is inside the core. This leads to the formation of micelles. So micelle formation facilitated by bile salts is very important for lipid digestion and absorption. So what is happening, if you see the picture here, you have on the left side fatty acid, right? It is the fatty acid with a non-polar tail and a polar head group. When you take a mixture of fatty acids and you put in water, what will happen? Micelle structures will be formed. Now how the micelle is shown in the picture in the slide. You see the second picture, that is the structure of a micelle. If you see, those are fatty acids, okay? Those black lines are the hydrocarbon chain of the fatty acid, which is hydrophobic, and those pink head regions are hydrophilic head groups, that is carboxyl groups. So when these fatty acids are mixed in water, what is happening? They are getting oriented in such way such that you see all the pink head groups, which are hydrophilic in nature, are projected outwards and they are interacting with the aqueous phase, whereas the non-polar tail groups or the hydrocarbon chains are getting projected inside. They are inside. If you see, they are inside, right? So this is how micelles are formed, that is to orient themselves. These fatty acids, when they are mixed with water, lipids when they are mixed with water, they orient themselves in such a way such that the hydrophilic groups will be exposed outside and they will be interacting with the aqueous phase, whereas the hydrophobic groups will be projected inward into the core, right? So what is the significance of this micelle formation? Micelle formation facilitated by bile salts is very important for lipid digestion and absorption. So these micelles are formed during lipid digestion and absorption, and bile salts are involved in the lipid micelle formation. Next, membrane bilayers. In case of biological membranes, a bilayer of lipids is formed orienting the polar head groups to the outer aqueous phase on either side and non-polar tails into the interior. The formation of a lipid bilayer is the basis of membrane structure called multilamellar membrane structure. So you have unilamellar, you have multilamellar. Unilamellar structure is single, okay, single layer is called unilamellar. Multilamellar is more than one layers is called as multilamellar, okay. So you can have a unilamellar or a multilamellar structures being formed. So in membrane bilayers, if you see in the picture, you see you have a bilayer structure wherein the hydrophobic tails are projected inward and the hydrophilic head groups are projected outward on either side of the membrane they are present. So on the either side they are interacting with the aqueous phase. In this way, membrane bilayers are also formed. So in aqueous phase, what all structures a lipid can take? It can take micelle structure, it can form membrane bilayers, and membrane bilayers, if you take the cell membrane is made up of membrane bilayers. So membrane bilayer gives a sort of structure to the cell and maintains the cell permeability and integrity. Next, vesicle. In cell biology, a vesicle is a structure within or outside a cell consisting of liquid or cytoplasm enclosed by a lipid bilayer. So what is vesicle? Just imagine. In micelle, what did we see? We saw hydrocarbon chains of the fatty acid are projected outward and hydrophilic groups are projected hydrophobic regions are projected inward and hydrophilic groups are projected outward, right? But in vesicle, what do we see is this vesicle has outside, this can be present within a cell, inside the cell, or outside the cell. A vesicle can be present inside a cell or outside a cell, and it consists of liquid or cytoplasm enclosed by lipid bilayer. So you will have a lipid bilayer will take a circular shape, and inside that lipid bilayer cytoplasm or some liquid will be present. That is the vesicle, okay. Vesicles form naturally during the process of secretion such as exocytosis and endocytosis. Secretion is exocytosis, taking up is endocytosis, okay. So during those processes vesicles are formed, and they transport of materials within the plasma membrane will also take place through vesicles. Alternatively, they may be prepared artificially, in which case they are called as liposomes. So if the vesicles are formed biologically inside us, we call them as vesicles. But when they prepare them outside, when we prepare them outside, then we call them as liposomes. Artificially prepared vesicles are liposomes. Simply putting like, we can say artificially prepared vesicles are liposomes. Now the membrane enclosing the vesicle is also a lamellar phase, similar to that of plasma membrane. So same like plasma membrane, the membrane of the vesicle is similar to plasma membrane, and intracellular vesicles can fuse with the plasma membrane to release their contents outside the cell. So for the purpose of endocytosis or exocytosis, the vesicles have to fuse with the plasma membrane, right? So here see, for the purpose of exocytosis, what will happen? Suppose you have to throw something out of the cell. So those some materials which have to be thrown out of the cell will be incorporated into the vesicles. The vesicle will get transported to the plasma membrane. The vesicles will fuse with the plasma membrane and it will throw that unwanted material out, right? Then what happens during endocytosis? Endocytosis, if the cell has to take some material inside, the plasma membrane will engulf that material. The plasma membrane will engulf that material, take it, and it will pinch out a vesicle inside the cell. In that way, vesicles are important in endocytosis and exocytosis. Vehicles can fuse with other organelles within the cell. So vehicles can also fuse with other organelles within the cell. A vesicle can also fuse with organelles within the cell. A vesicle released from the cell is known as extracellular vesicle. Vesicles perform a variety of functions. Because it is separated from the cytosol, the inside of the vesicle can be made to be different from the cytosolic environment. See, the vesicles can contain cytosol, and the vesicles cannot, you can remove the cytosol portion also. That is, you see, we also discussed in the previous point that vesicles can fuse with organelles, right? So, the inside portion of the vesicle can be cytosol portion, or it could be separated from the cytosol. You see, a vesicle is an enclosed structure, right? So whatever material is present inside the vesicle may not be present in the cytosol, right? So you can separately see the material present inside the vesicle will be different from the cytosol. Once it gets separated, it will be different from the cytosolic environment. For this reason, vesicles are a basic tool used by the cell for organizing cellular substances. So vesicles functions, they are involved in metabolism, they involved in transport, buoyancy control, temporary storage of food and enzymes. They can also act as chemical reaction chambers. So this is about vesicles, how they are formed, what are their functions. Next, coming to liposomes. So liposomes are nothing but, I already told, they are artificially produced or synthesized vesicles. So liposomes, they are produced when amphipathic lipids in aqueous medium are subjected to sonification. So they have intermittent aqueous phases in the lipid bilayer, and liposomes in combination with tissue specific antigens are used as carriers of drugs to target tissues. If there is only one phospholipid bilayer, they are called as unilamellar liposome vesicles. Okay, so artificially prepared vesicles are liposomes, right? So similar to vesicles, their structures will be, and they are prepared when amphipathic lipids in aqueous medium are subjected to sonification. The sonification is a process wherein the component, that is in this case the aqueous phase containing amphipathic lipids, is subjected to very high vibrations, okay, very high agitations. You have a sonicator, in that you will put, and the process vibrations, because of this huge vibrations, that mixing will take place and these liposomes will be formed. Okay, so they have intermittent aqueous phase in the lipid bilayer. If you see the structure here, you will understand in this slide. You see the left one is the unilamellar and the right one is the multilamellar. So you see the difference. In unilamellar you have only a single bilayer, circular, it is circular in shape, it is lipid bilayer, right? And inside the blue color, that is the environment, liquid environment, or the whatever aqueous or liquid environment present inside the liposome. If you see the multilamellar, you see there are here if you see there are three layers exactly. First layer, second bilayer and third bilayer. So this is multilamellar. In multilamellar, between each layer you will have a environment. You have an aqueous environment or a liquid environment. So what are the uses of these liposomes which are artificially prepared? So liposomes in combination with tissue specific antigens, okay, in tissue specific antigens act as carriers of drugs to target tissues. So if you want to do some drug delivery experiments, okay, so what you do, you make these artificial liposomes, you make them in such a way such that the drug is incorporated into that inside environment. Whatever liquid environment is present, you have to incorporate the drug into it, and that liposome containing the drug is induced to that particular model of organism or an animal, and the drug will get delivered. So liposomes act as carriers of drugs to target tissues, and they could be unilamellar as seen in the picture, in the left picture, or multilamellar as seen in the right picture. Okay, so that was about behavior of amphipathic lipids in aqueous solutions. We have seen formation of micelles, bilayers, liposomes and vesicles. Next, coming to fluid mosaic model. Fluid mosaic model proposed by Singer and Nicholson is a more recent and acceptable model of membrane structure. Thickness of 5 to 8 nanometers. So according to fluid mosaic model, the membrane structure, the membrane thickness is 5 to 8 nanometers and composed of a lipid bilayer The hydrophobic regions of the lipids, the hydrophobic, that is non-polar regions of the lipids, face each other at the core of the bilayer, and the hydrophilic regions face outward. Okay, similar, you see any molecule which is mixed with the aqueous solution, it will acquire this kind of orientation only. That is, hydrophobic regions will be projected interior and hydrophilic regions will be projected on the exterior or outward. Globular proteins are irregularly embedded in the lipid bilayer. So in fluid mosaic model, you will have a phospholipid bilayer. Apart from the phospholipid bilayer, you will have proteins embedded in them. In the lipid only, proteins also will be present. Now membrane, these are called as membrane proteins because they are present in this phospholipid bilayer membrane, right? So since they are present in the membrane, such proteins are called as membrane proteins, and there are two types of membrane proteins: extrinsic proteins, intrinsic proteins. So extrinsic membrane proteins are also called as peripheral membrane proteins because they are present on the periphery only. These are loosely held to the surface of the membrane and they can be easily separated. So what are the characteristics of extrinsic membrane proteins? They are loosely bound to the surface of the membrane, and since they are loosely bound, they can easily be separated by centrifugations. Examples are cytochrome C of mitochondria. Example is cytochrome C of mitochondria. Next, intrinsic membrane proteins are also called as integral membrane proteins, are tightly bound to the lipid bilayer, and they can be separated only by the use of detergents, organic solvents. You see the difference? Extrinsic, loosely bound. Intrinsic, tightly bound. Extrinsic, easily separated. Intrinsic proteins cannot be separated that easily, and they can be separated only by the use of detergents, organic solvents. Examples are hormone receptors, cytochrome P450. These are intrinsic membrane proteins. Next, the membrane is asymmetric due to the irregular distribution of proteins. We use the term asymmetric, means if you cut the lipid bilayer in the middle, suppose there is a lipid bilayer, the plasma membrane, not lipid bilayer, I'm sorry. Let us take, here we are discussing about plasma membrane or cell membrane, right? So if you take the plasma membrane and cut it in the middle, you will not get same type of fatty acids or same type of proteins on either side. That symmetry will not be present. So you can say the membrane is asymmetric due to irregular distribution of proteins. Whatever proteins are present, some may be present outside, some may be present inside, some may be traveling along the whole of the plasma membrane, right? So if you cut it, the portions will not be same. So membrane is asymmetric due to irregular distribution of proteins. The lipid and protein subunits of the membrane give an appearance of mosaic or ceramic tile. So that is the reason it is called as fluid mosaic model, because when you see the picture, if you see in the slide, you see the picture of plasma membrane, you see there is a mosaic pattern, right? There is a mosaic pattern. So lipid and protein subunits of the membrane, the arrangement will give an appearance of mosaic or ceramic tile. So that is the reason they have given the name fluid mosaic model. Unlike a fixed ceramic tile, the membrane freely changes. So if you take normal tiles in your house, they are fixed, they will not move. But your plasma membrane, though it gives that mosaic pattern look, The membrane can freely change. Hence, the structure of membrane is considered as fluid mosaic. Since it can change its structure a little bit, it's called as fluid mosaic. Now, you see in the picture, you can observe the mosaic nature of the plasma membrane, and you see that is the phospholipid bilayer. Those green structures and those black threads, that is the phospholipid bilayer. And you see the proteins, you see peripheral proteins, you see only they are attached to surface, whereas integral proteins, they are embedded into the membrane, they are crossing the membrane. So if they are crossing the membrane, if they are into the membrane, you call it as integral protein or intrinsic protein. If they are just attached to the surface, you call them as peripheral proteins or extrinsic proteins, right? So this is the structure of the fluid mosaic, nature of fluid mosaic model of the plasma membrane. So that's it for today's class. So in today's class, we have learned two topics. One is behavior of amphipathic lipids in aqueous solutions. What all structures it will take when lipids, what all structures lipids will take when they are put in the aqueous solution. They are micelle structure, lipid bilayers, vesicles and liposomes. And we have also discussed about fluid mosaic model of plasma membrane. Thank you.